Hello friends, today we are going to discuss about the pentose phosphate pathway, or it is also known as the HMP path. Initially we have to discuss about the glycolysis and the phases of glycolysis. So the number of cells, or each and every cell, they have the alternative pathway for the breakdown of the glucose. Basically glycolysis, they will be converts the glucose into the pyruvate and the net gain of 2 ATPs and also the 2 NADH. Then also the specific pathways are required for the synthesis of the different types of the ribonucleotides also. Ribonucleotides most of the times nucleic acids, for example RNA, DNA. So each and every cell they have to carry out the specific alternative pathway for the synthesis of nucleic acids mostly because nucleic acids are 5 carbon compound nucleic acid that contain the specific sugars, and these sugars are the 5 carbon 5 carbon molecule. So the specific pentose phosphate pathway, it is also known as a phosphogluconate pathway. It is also known as a phosphogluconate pathway. Then also known as a pentose cycle, hexose monophosphate pathway, or also known as a Warburg-Dickinson-Limpan pathway. That is a Warburg-Dickinson and Limpan pathway. So these are the other names for the pentose phosphate pathway. So the definition is that the process which brings about oxidation and decarboxylation at the carbon first of glucose 6 phosphate, reducing NADP to NADPH and producing the pentose phosphates. In this specific pathway, the process which brings about the oxidation and decarboxylation reactions are carried out in this specific pathway at the position number first of the specific glucose, that is a glucose 6 phosphate. In case of the glycolysis, glucose get converted into the glucose 6 phosphate. After that, the glucose 6 phosphate get converted into the fructose 6 phosphate, and this fructose 6 phosphate get converted into the fructose 1,6 bisphosphate. But in this pathway, only the glucose get converted into the glucose 6 phosphate, and then the specific oxidation and decarboxylation reaction will be carried out, and they can synthesize, or they will specific types of the 6 carbon molecule, they will be converted into the 5 carbon, and it is known as a pentose phosphate. So it is because of this it is known as a pentose phosphate pathway. So occurrent, it is most of the it is occurs in a cytosol or cytoplasm of each and every bacterial cell. It is not only carried out in case of the bacteria, it is also carried out into the human and animal cells. So most of them distribution: liver, lactating mammary glands, adrenal cortex, gonad, adipose tissue, erythrocytes, lens, and cornea. There are different locations where the specific types of the pathways are carried out. So the biomedical importance is that it has two main functions. It provides the NADPH and also provides the pentoses. It provides the NADPH and also the pentoses. Through the there is oxidation of glucose, but it is not meant for the energy. Here is a specific reaction in which the oxidation of glucose is carried out. It is not meant for energy. It is not a pathway they will be carried out for the energy generation. So energy generating pathway, it is mostly glycolysis. So it is an also path. It is an only pathway. Most of the times it is carried out for the formation or synthesis of pentoses. Metabolic pathway. This is a metabolic pathway. It is carried out into the two important phases. One that is the oxidative phase, and second one that is a non-oxidative phase. This is a specific PP path or pentose phosphate pathway. It is carried out in a oxidative phase and non-oxidative phase. In this specific phase, that is stage 1, that is oxidative phase, when the glucose get converts into the glucose 6 phosphate, when the specific cell they will be carried out the pentose phosphate pathway. At that time, this glucose 6 phosphate get converts into the 6 phosphogluconolactone. The glucose 6 phosphate get converts into the 6 phosphogluconolactone, and the specific enzyme responsible for this is glucose 6 phosphate dehydrogenase. Glucose 6 phosphate dehydrogenase, and they will be convert the NADP into the NADPH plus H plus, and the cofactor is Mg double plus. So the specific glucose 6 phosphate when converts into the 6 gluconolactone with the help of enzyme glucose 6 phosphate dehydrogenase, and the specific NADP they get converts into the NADPH plus H plus. Then this 6-phosphogluconolactone, it converts into the 6-phosphogluconate. It is an acetate, or the specific types of the gluconolactone get converts into the gluconate. The enzyme responsible for this reaction, it is 6-phosphogluconolactonase. 6-phosphogluconolactonase, and the specific types of the breakdown of H2O, and H2O they converts into liberation of H plus. Then cofactor is also Mg double plus. 6-phosphogluconolactone get converts into the 6-phosphogluconate. The specific as the stage 1 is further continued. The 6-phosphogluconate get converts into the 3-keto 6-phosphogluconate with the help of enzyme 6-phosphogluconate dehydrogenase. Here in this first step, the specific enzyme, that is a glucose 6-phosphate dehydrogenase. In this specific types of this in this reaction, glucose 6-phosphate get converts into the 6-phosphogluconolactone. In this specific stage also, the 6-phosphogluconate get converts into the 3-keto 6-phosphogluconate, and the enzyme is 6-phosphogluconate dehydrogenase. The specific NADP get converts into the NADPH. Cofactor is same, that is a Mg double plus. The specific 3-keto, the 3-keto means at the position number third, carbon, the ketonic group is present, that is a C double bond O. 6-phospho, that is a 6-phosphate, or the phosphate group is present at the position number 6. The specific 6-phosphogluconate get converts into the D-ribulose 5-phosphate plus carbon dioxide. When 3-keto 6-phosphogluconate converts into the D-ribulose 5-phosphate. D in the it is a dextrorotatory 5-phosphate at the position number 5. If the phosphate group is present, and the specific carbon, it will be removed from the specific 6-carbon molecule, or that is a glucose from the glucose 6-phosphate, or it is a specific glucose 6-phosphate get converts into the 3-keto 6-phosphogluconate, and this 3-keto 6-phosphogluconate get converts into the D-ribulose 6-phosphate plus liberation of one molecule of CO2. Then it is an oxidative phase. The first one phase, that is a oxidative phase, and second one the phase, that is a non-oxidative phase. In this phase, interconversion of pentoses. Pentoses will be interconverted into the one another. The D-ribulose 5-phosphate get converts into the D-ribose 5-phosphate. The D-ribulose, it is a specific ketonic form. This ketonic form, it is converts into the aldolase form. Then D-ribose 5-phosphate, the phosphopentose isomerase. The isomerization reaction, D-ribulose 5-phosphate to D-ribose 5-phosphate. D-ribulose 5-phosphate to D-ribose 5-phosphate enzyme, that is a phosphopentose isomerase. The D-ribose 5-phosphate get converts into the D-ribose 1-phosphate at the position number 4, that is a phosphate group, and also D-ribose 1,5-diphosphate. So the specific, there are the two parts, D-ribose 5-phosphate, otherwise the also this specific D for ribose 5-phosphate get converts into a D-ribose 1-phosphate or D-ribose 1,5-diphosphate. The two phosphate molecules are present at the position number 1st and position number 5th. Then in case of the stage 2nd, the D-ribulose 5-phosphate get converts into the D-xylulose 5-phosphate. D-ribulose 5-phosphate get converts into the D-xylulose 5-phosphate, and the enzyme, that is phosphopentose epimerase. The phosphopentose epimerase. The ribulose get converts into the xylulose. It is a epimerization reaction, the change in the position of the hydroxyl group. The specific ribulose, D-ribulose 5-phosphate get converts into the D-xylulose 5-phosphate. There is a change only hydroxyl group because of the enzyme phosphopentose epimerase. Phosphopentose epimerase. Then this specific types of the D-xylulose 5-phosphate get they will be moves into the second reaction. The second stage is a conversion of PP to hexose 5-phosphate. The stage second into the stage second. The first one, that is a interconversion of pentoses, and in this second, or B, conversion of PP, that is a pentose phosphate to hexose phosphate. Conversion of PP or pentose phosphate to hexose phosphates. The two particular enzymes are required, that is a transcatolase and transaldolase. Transcatolase and transaldolase, they are the two important enzymes. These are required for the conversion of PP to hexose phosphates. The transcatolation, it is that xylulose 5-phosphate plus xylulose 5-phosphate plus ribose 5-phosphate. They both are pentose phosphates. This is xylulose, it is also pentose, ribose is also pentose sugar. But xylulose it is a ketonic sugar and ribose it is a aldose sugar. So the trans catalyze it is an enzyme act on its specific xylulose plus ribulose 5-phosphate. Then this pseudoheptulose, that is a pseudoheptulose pseudoheptulose 7-phosphate plus glyceraldehyde 3-phosphate. Total number of carbons, that is a 5 from xylulose, 5 from ribose. Total 10 carbons. Into that 7 pseudoheptulose 7-phosphate. It is a 7 carbon sugar plus glyceraldehyde 3-phosphate. It is a trans ketolation. The specific types of the ketonic group from the xylulose, they will be converted to the xylulose to pseudoheptulose. There is a conversion of specific transcatolic or the specific types of the one ketonic sugar and one aldehydic sugar, they will be converts into the two different sugars, one that is a pseudoheptulose and second one that is a glyceraldehyde 3-phosphate. Then transaldolation. Pseudoheptulose Pseudoheptulose 7-phosphate plus glyceraldehyde 3-phosphate, when interconverted reacted with one another with the help of enzyme transaldolase, they can form the fructose 6-phosphate plus erythrose 4-phosphate. Fructose 6-phosphate, it is a specific sugar. It is a sugar that is a ketonic one. The fructose 6-phosphate is a ketonic sugar and erythrose 4-phosphate it is a aldehydic sugar. So the when the pseudoheptulose 7-phosphate plus glyceraldehyde 3-phosphate reacted with one another with the help of in the presence of enzyme transaldolase, they can form the fructose 6-phosphate plus erythrose 4-phosphate. Then transcatolation. Xylulose 5-phosphate plus erythrose 4-phosphate, they can form the fructose 6-phosphate plus glyceraldehyde 3-phosphate. Also, this glyceraldehyde 3-phosphate, they will be interconverted into the dihydroxyacetone phosphate plus glyceraldehyde 3-phosphate. Then also interconverted into the fructose 1,6-bisphosphate. Also, this fructose 1,6-bisphosphate, they get converts into the fructose 6-phosphate. This fructose 6-phosphate get converts into the glucose 6-phosphate, and the recycles the pathway. Further, this glucose 6-phosphate, they will be moves into the first step or first stage of this specific pentose phosphate pathway, that is a glucose 6-phosphate. In this last stage, the specific types of the sugars, when xylulose 5-phosphate plus erythrose 4-phosphate in the presence of transcatolase enzyme fructose 6-phosphate plus glyceraldehyde 3-phosphate, then this glyceraldehyde 3-phosphate get converts into the dihydroxyacid DHAP plus glyceraldehyde 3-phosphate, then interconversion of this into fructose 1,6-bisphosphate, then fructose 6-phosphate to glucose 6-phosphate, and also the specific glucose 6-phosphate will be recycled into the same reaction or into the same pentose phosphate pathway. Here the completion of hexose monophosphate shunt or pathway, or it is also known as a pentose phosphate pathway.